Good morning. Someone will shout if you don't hear me, right? I don't hear any feedback, so. Thanks very much to the organizers for putting together this really excellent program, and also for inviting me to share some of the work that we're doing within my group at the MPI of Biophysics in Frankfurt. So within my group, we our mission, let's say, is to determine structures and understand mechanisms of different redox proteins that play important roles in shaping life, but in particular in shaping natural environments and our climate. And as we try to understand these systems better, of course, we have to be able to ask mechanistic questions at a structural level. And for redox proteins, that means being able to understand how the structure of the protein actually changes with redox status. And so some of the projects that we're working on have kind of pushed us to bring together a little bit of a toolkit. And that's what I want to talk about just kind of briefly in case that's useful for anyone else's work. So the kind of questions that we deal with are on a sort of spectrum between relatively simple and a little bit more complex, where simple is something like just working with an oxygen-sensitive complex or preparing a stably reduced state. And where complex moves more into trying to hit particular states that are already defined by spectroscopic studies, but where we would like a structural understanding of those studies. And so the tools that we brought together are relatively simple, but they allow us to answer quite a few new questions that we wouldn't otherwise be able to answer. And so we start by just putting a Vitrobot into a glove box. That's a pretty simple thing to do. The only things to kind of pay attention to there are heat dissipation from the Vitrobot and humidity, and then keeping the oxygen low in the liquid nitrogen before we take it into the anaerobic tent. And so that's, if anyone's kind of interested in trying that at their home institute, feel free to come and chat with me after the talk. But that already allows us to answer a lot of questions that we otherwise wouldn't be able to, because we can stably reduce a complex, and we can also work with complexes that we know will break down in the presence of oxygen. And when we add to that a gas mixer, so that doesn't, I don't know if that's obvious what that is, but that's actually a set of mass flow controllers that allow us to make mixtures of gases, because a lot of the proteins that we work with cycle different or use gases as substrates. And so that's also allows us to hit particular states. And then toward the slightly more complicated end, but still a relatively simple tool, is solution potentiometry. So this is actually a figure from a paper of John Wright when he was in Maxi Rüssler's group at Imperial. And they use this technique to prepare samples for EPR spectroscopy. But we can measure the potential of a solution of purified enzyme and similarly apply it to a grid. So that's just a 2-grid setup where you have one working electrode and one reference electrode in solution with a potentiometer. So again, if there's someone who's kind of interested in that, feel free to chat with me afterward. So I suppose the tools are only interesting if they allow us to ask or answer interesting biological questions, and we're sort of slowly building up the projects that move across this range, although some of the questions we'd like to ask really just need the simpler stuff like stable reduction. And so now I'd like to tell you about a couple of those different projects that we worked on and try and kind of tell you the stories of those proteins and what we've been able to learn about them from the combination of these tools and, of course, single particle cryo-EM. The first is about methanogenesis. So hydrogenotrophic methanogens take CO2 and they reduce it and fix it. And you don't really need to worry about the details of the cycle. All you need to know is that CO2 is entering here and being reduced and fixed, and then moving through this whole cycle, becoming successively more reduced until that carbon is released as methane. And in that process, an ion gradient is built up that actually gives these organisms the energy that they need to survive and grow. And that's quite an amazing reaction, because the electron donors that methanogens have at their disposal for this reaction, for the CO2 reduction that then makes the fixation favorable, are not sufficiently reducing to actually drive that reaction forward. So they have hydrogen and formate. And in order to kind of deal with that problem, in this cycle, methanogens also have a reaction called electron bifurcation. And so the electrons from hydrogen or formate come into this electron bifurcating complex, and a pair of electrons basically enters the bifurcating site, and the reducing power of those two electrons is split to generate a more strongly reducing electron and a less strongly reducing electron. And the more strongly reducing electron is then used to make this CO2 reduction and fixation favorable. So that's quite a neat reaction, but there are lots of details about that that are unknown. Furthermore, the idea that's sort of shown in this scheme is that these two reactions are separate, but a postdoc in Seigo Shima's group at the MPI of terrestrial microbiology in Marburg was able to purify a complex that actually brings both of these two activities together. And so we worked together with them to solve the structure of this complex. Olivia Pyle-Gardner is a PhD student in my group who worked on this project. And there was a fair bit of kind of image processing, because when we first looked at the complex, we wondered where most of it was. So we were just really seeing a very small part of what we expected to be there on the basis of SDS page and mass spec. And that's because there's a huge amount of conformational flexibility. That's related both to the complex being very fragile and to a conformational change that I'll tell you more about in a minute. But by quite a lot of masking, Olivia was able to eventually generate a composite map of the full complex. As you can see, well, maybe you can't see, but in this particular complex, we have basically a dimer. So we have two of every chain. And when we look at this structure, we basically have three take-home messages about the process of electron bifurcation and how that is able to drive CO2 fixation and methanogenesis. The first is related to substrate flexibility. So here in the electron donor arm, we have the formate oxidation site. So the formate dehydrogenase site that actually uses formate in order to generate these electrons for bifurcation. But it turns out that there's an additional site here that's able to oxidize an organic cofactor called F420H2, which within the cell is then reduced by hydrogen. So this structure gave us a new understanding of how these organisms can actually use both hydrogen and formate, even if they only have this one particular complex. And the second take-home message was related to electron transfer through the complex. So previously, it wasn't clear how electrons are actually moving through the complex. But by looking at two different conformational states, we got a new insight into that. So it seems that electrons are coming from the electron donor arm, or what we're now calling the mobile arm, from either this formate dehydrogenase site or from the F420H2. And from there, they seem to be able to make the jump to the bifurcating site, which is a flavin cofactor found within the heterodisulfide reductase. But if we then move to the second conformation that we observed, we can understand how electrons can move on from that. So following bifurcation, the less strongly reducing electron goes for heterodisulfide reduction, and the more strongly reducing electron is used to reduce this particular shuttle cluster. And now moving back to the first conformation, we're able to see how electrons can move onward. And the third take-home message is related to how these two complexes are linked together, because as it turns out, they not only form a supercomplex, but they in fact form an electrically connected supercomplex, which allows these very precious electrons generated by electron bifurcation to be transferred directly via a polyferredoxin subunit all the way to the CO2 reduction site. As it turns out, that polyferredoxin subunit is only one of several types of this protein of polyferredoxin that's expressed by methanogens at relatively high level. And so it seems that this might actually be a more general theme that hasn't been explored so far, because these complexes tend to be quite fragile and extremely large. There is still a problem, which is related to electron transfer in this step. So here we have a 2-iron cluster, and here we have the bifurcating flavin, and the distance there is still too long for electron transfer. But moreover, there's a kind of mechanistic problem here, because what we know about electron bifurcation is that we should have a cofactor that has a high energy intermediate state. So the cofactor, once one electron is lost, the other electron should come off with quite a lot of energy, and that's how the bifurcation actually occurs. And in most bifurcating systems, that's bypassed in the reductive direction by having a hydride transfer. So you transfer 2 electrons and a proton at the same time, and therefore in the reductive direction, you bypass that high energy intermediate. But here we have electrons coming from all the way out here at the active sites down a chain of iron sulfur clusters, and therefore more or less by definition one at a time. And so this is a kind of unsolved mystery of this system. But one of the conformational states that we solved gives us some new insight in the sense that we see all of these very well conserved thiol residues lying between that 2 iron cluster and the bifurcating flavin. And it turns out that this flavin binding domain is homologous to thioredoxin reductases, which catalyze a reduction of a disulfide bond via a covalent intermediate with a flavin. And so that's our best hint at what might be going on, that these complexes might actually be catalyzing that reaction in reverse. But we still have a lot of work to do to see whether that is in fact the case. I spoke about the fact that we had a lot of conformational flexibility, and some of that was related to these 2 conformational states of the mobile arm. But some of that was also related to this 2D class that we saw in our initial data set that didn't look like any of the other 2D classes that we observed. And we wondered whether that could be a higher order complex. And so I asked Tomohiro to send us this little shoulder in yellow, which was only 9 microliters of 0.1 mg per mil, but cryo-EM can sometimes be very kind, and that was enough to generate a few more 2D classes and eventually solve a structure of this larger hexameric complex that effectively is made up of 3 copies of our dimer. And what you can see here is that the polyferredoxin subunits, which are shown in green and teal, that conduct electrons from the electron bifurcating site to the CO2 reducing site, actually form a kind of belt around the complex so that electrons can be conducted between these different units. So I think in the interest of time, I'll probably just move through the summary, but the next story that I wanted to tell you about is the formate hydrogen lyase complex. So this is a complex that sits in the membrane of E. coli, and it catalyzes a reaction where it oxidizes formate and uses the electrons from that oxidation in order to generate hydrogen. So that's quite an interesting reaction, and it turns out that this is much better at making hydrogen than other related hydrogenases. It's also possible to force this reaction to work in reverse. So work done in the group of Frank Sargent show that by putting this complex under a pressure of H2 and CO2, it's possible to drive CO2 reduction to formate. But the reason that we're really interested in this complex is hinted at by the shape of the complex, which you may recognize bears some similarity to complex 1 with this L-shaped form. And it turns out that these types of hydrogenases, the type 4 membrane-bound hydrogenases, are the ancestors of respiratory complex 1. And based on sequence similarity, it seems very likely that this whole complex 1 superfamily has a conserved mechanism of coupling the redox reaction in the soluble arm with proton translocation across the membrane arm. There are 4 different groups in this superfamily, all of which reduce different substrates at an active site that's equivalent to the quinone reducing site in complex 1, but they all seem to work in the same way, as far as we can see from sequence and now from structure. But what we don't know is what that core mechanism actually is. And so we think that this is an interesting system for basically taking a very simple look at that mechanism, because we don't have the complications of active and deactive transitions. We don't have a specific quinone chemistry that might be going on. We really have a very simple substrate. We're just reducing protons and releasing hydrogen. And so Ralph Steinhöfer, there we go, in my group set about trying to solve a structure for this complex. And Ralph sometimes feels a little bit embarrassed when I show this micrograph, because it's true that it doesn't look all that good, but it's part of the journey. And so when in some of the first purifications, it was clear from the SEC profile that not everything was really working perfectly. We have two pretty strong peaks, one of which is missing some of the subunits. But in any case, it was possible to use these first images to generate an initial structure. And when we were starting to build a model into that structure, one of the things that became quite apparent very early on was that some of the redox cofactors that we expected to be there didn't show a strong density like we would expect them to. So neither the nickel iron based hydrogenase active site or the next nearest iron sulfur cluster showed a strong density. And so it was essential to kind of go back and figure out what was going wrong. And what seemed most likely was that oxygen was in fact inactivating this site, which we think is really essential to the coupling mechanism. And so Ralph got to work setting up the purification completely anaerobically. And immediately the SEC profile started to look better. But when he solved a structure of this, we did in fact see the densities that we were expecting at the cofactors, but we saw that we were missing an entire subunit. We were missing the formate dehydrogenase subunit here at the top. And Ralph worked very hard to try and eliminate any possible differences between the aerobic and the anaerobic prep, working on the assumption that this was some kind of an artifact, until we eventually convinced ourselves that it may have a regulatory function. And I can tell you more about that in a little bit. But just to kind of summarize the differences between these preps, not only did we maintain the cofactor in the anaerobically isolated state, but we also had much better order in the flexible loops that connect the soluble and the membrane domains, and we retained the membrane-bound subunits. And then the final test, of course, is the activity. So the hydrogenase activity of this anaerobically purified complex is substantially higher than the aerobically purified. And so that gives us a lot of confidence that that's basically the way that we should be looking at this complex. When we look at the cofactors, we had a number of iron sulfur clusters that link the molybdenum-based formate dehydrogenase active site with the nickel iron hydrogenase active site. But we had an additional metal ion here that we didn't actually anticipate being there, so that hadn't been predicted before. And it turns out that that sits really close to the interface between the complex and this formate dehydrogenase subunit. So I can tell you more about that in a little bit. But one of the reasons that we were studying this is because of the similarity to respiratory complex 1. And so what about similarities and differences to complex 1? The first is a really large difference, and this was already shown by Michael Adams' group with an archaeal version of a similar enzyme. And what's really interesting is that if you actually align the complexes on the basis of the antiporter-like subunit, the whole rest of the complex has actually shifted around to the other side of the antiporter-like subunit. So if you want to put it another way, you have an 180-degree rotation in the plane of the membrane of the antiporter-like subunit. And so what this suggests to us is that there's an extremely simple coupling mechanism if it's able to be preserved through that 180-degree rotation. Another strong and even strikingly strong similarity between the complex and complex 1 is this set of loops that connect the soluble domain with the membrane domain. These show, even where there's relatively low sequence homology, they show a striking amount of structural similarity. And I just want to point out this beta-1, beta-2 loop. So in complex 1, the beta-1, beta-2 loop is really close to the quinone reducing site, and it hosts a histidine that seems to be the most likely proton donor for quinone reduction. And it turns out that in hydrogenases, that histidine is replaced by an acidic residue that could also donate protons. And spectroscopic studies of soluble hydrogenases have actually shown that that particular acidic residue is the proton donor to the active site. So if we get a proton moving from the nickel iron active site, first it goes via this terminally coordinating cysteine, and from there it moves on to that glutamate, which is the equivalent of the histidine on the beta-1, beta-2 loop. And so that's a really striking similarity for two complexes that diverged several billion years ago. And here we even have a case where we have a substitution, but a retained function. And so this points to a really fundamental importance for substrate proton movement in the coupling mechanism of complex 1 and its relatives. But how we actually protonate that glutamate seems to be strikingly different to soluble hydrogenases. So there are a number of different pathways that have been mapped out from crystal structures, very high resolution crystal structures of soluble hydrogenases. And those are all knocked out in FHL by mutation to non-proton conducting residues. And so the question is, how do you actually protonate that residue? And there we have another important similarity to complex 1, because this beta-1, beta-2 loop sits at the top of a large hydrophilic channel that extends all the way from the nickel-iron site, or from near the nickel-iron site, deep into the membrane. This nickel-iron site, by the way, is in the same position where you would have a quinone-reducing site in complex 1. And from everything we know, that seems to be the coupling site across this whole superfamily. And so it points to potentially a different route for protons to actually reach that beta-1, beta-2 loop that actually goes via the membrane. And when we map different site-directed mutants onto this structure, it's interesting to note that mutations all the way down here, for example, this E138, this is work done in the group of Frank Sargent at Newcastle University. And it turns out that mutation, for example, of this glutamate all the way deep down in the membrane effectively knocks out hydrogen production activity several nanometers away. And so again, this is pointing for us to a very fundamental importance of substrate protons in the coupling mechanism within the complex 1 superfamily. And so, of course, future studies are needed to really understand exactly how that works, but we still think that this is giving us some important new hints. I mentioned that we always have this formate dehydrogenase falling off. And initially we thought, we're just doing something wrong. We'll kind of keep, working away at it. But when we started to build the model of the full complex and we found this additional metal ion, it became clear to us that it's possible that there's a structural mechanism for actually controlling the association and dissociation of the formate dehydrogenase. Because we have an ion here, we've labeled it as an iron, but we actually don't know, which is a bit of a problem, I think, for cryo-EM in general, that sometimes we don't have a good way of definitively assigning unknown densities. But we've worked on the assumption that this is an iron because it has 4 cis coordination. And this iron actually turns out to sit right in a conserved positively charged patch that interacts with a negatively charged patch on the FDHF. And so when we look at that, the sort of running hypothesis is that under more reducing conditions, the overall charge of this area becomes less positive, and therefore the interaction with the FDHF subunit is less strong. And that's why we're losing the FDHF subunit under reducing conditions. This could have a function if we think that the complex is actually supposed to work in the formate oxidizing direction in cells, then we could imagine that this helps to reduce the reverse rate by causing a reduced complex to not associate with the formate dehydrogenase in order to not be able to use those electrons for CO2 reduction. Again, in the interest of time, I think I'll kind of just breeze through the summary, but I'd like to thank Ralph and Olivia, whose work I've shown today, as well as Max, Pablo, and Victor in my group, who've all contributed to kind of bringing together these different tools for understanding redox proteins, as well as our really excellent central electron microscopy facility at the Institute, and our collaborators at Newcastle, at the MPI for terrestrial microbiology, and at the Philipps University in Marburg, as well as the DFG and the Max Planck Society for funding. Thanks very much for your attention.